Petunia plants are being cultivated in a field at the University of Rostock for an experiment dealing with cross-pollination. The petunias with white blossoms are conventional plants. The ones with purple blossoms have been genetically modified and have a special characteristic. The new genetic information was not inserted into the cell nucleus, but into the plastids. Plastids are small subunits within cells and possess their own DNA. Since the genetic information of the plastids is not transported by pollen, this information remains genetically sealed within the plant. This way, the spread of newly inserted genes may be prevented. With petunias as the model plant, field trials are now testing if it actually is the case that no plastid DNA is transported by pollen. Patricia Horn and her colleagues are at the field today to pollinate chosen plants by hand. White, conventional plants with flower buds that are still closed are chosen and marked. In their case, one can be absolutely sure that they have not been already pollinated by the pollen from other plants. The pollen sacs in the blossoms are removed.
I'm
castrating the flowers. What I mean is that I open them first and take out the pollen containers in order to avoid that they eventually ripen and lead to self-pollination in the flower. Blossoms also are chosen that have ripe pollen that can be used for pollination. The purple petals are removed and the pollen is carefully dusted onto the stigma of the white flowers. Today, seed capsules also are being collected. They contain large numbers of tiny seeds. In the lab, these capsules are opened and the seeds are separated from the rest of the capsule. A defined quantity of seeds is then sown on a nutrient base. In the greenhouse, in darkness and at a temperature of 18 to 24 degrees, it takes 2 weeks for them to grow into sprouts. They are then carefully harvested. Then, with a special color test, one may have the first hint of whether the sprouts contain the new genetic information or not. In this step of the project, we check, of course, each individual sprout and look at whether we find a blue coloration or not. If we find a blue coloration, then this is a sign that the improbable event has occurred, that is, that a plastid transfer has occurred through pollen. With the precision of a watchmaker, the sprouts are placed one at a time on paper and are inspected with a magnifying glass. Blue colorations are noted, and the sprouts are frozen in order to undergo DNA analysis later.